Welcome to the CRISPR in a Box educational instructional video where you will have the opportunity to edit DNA. In this kit, you will find the tools to complete the in vitro gene editing reaction, isolate DNA via a mini prep, and perform a double restriction digestion. But before we get into the in vitro gene editing reaction, there's a few steps that can be prepared ahead of time. So you can prepare LB agar plates and LB broth up to a week in advance. So to prepare the LB agar plates, you need to add X-Gal, IPTG, and carbenicillin. So you'll start by using a 1 liter glass bottle, add 500 milliliters of deionized water, and the entire bottle of agar powder. Autoclave the media at 121 degrees Celsius for 30 minutes with 15 psi. Then you'll cool the bottle to approximately 60 degrees Celsius and add the following reagents: 500 microliters of X-Gal, followed by 500 microliters of IPTG, and 500 microliters of carbenicillin. You will slowly mix this and then pour 20 to 25 milliliters of media per 100 millimeter plate, and then allow this to cool and store at 4 degrees until needed. Now to prepare the LB broth, you will take a 250 milliliter glass bottle and add 80 ml of deionized water and 2 capsules of LB broth. Autoclave the media again at 121 degrees Celsius for 30 minutes with 15 psi. Allow it to cool while swirling occasionally and store at 4 degrees until needed. So the day before the in vitro reaction, you need to start your overnight DH5 alpha E. coli culture 16 to 20 hours before preparing your competent cells. You will reconstitute your lyophilized DH5 alpha E. coli by adding 200 microliters of ultra-pure water. You will take 1 milliliter of the LB broth to a round bottom tube, and then add 20 microliters of reconstituted DH5 alpha E. coli to the tube. This culture then incubates at 37 degrees with shaking for 16 to 20 hours. On the day of the in vitro reaction, you need to prepare your competent cells. Prepare a 125 milliliter Erlenmeyer flask by adding 16 milliliters of Zymo broth. To the broth, you will then add 160 microliters of overnight culture grown from the day before and swirl to mix it. So grow the culture at 37 degrees with shaking until an optical density of 0.4 to 0.5 has been reached, or approximately 3 to 5 hours. While the cells are growing, you will pre-chill the 2x wash buffer, 2x competent buffer, and dilution buffer on ice prior to using in the preparation. You will then dilute the 2x wash buffer and 2x competent buffer to a 1x concentration, using the dilution buffer. To a 1.5 ml tube, add 600 microliters of dilution buffer to 600 microliters of 2x wash buffer. And now do the same with another 1.5 ml tube, add 600 microliters of dilution buffer to 600 microliters of 2x competent buffer. We highly recommend using a room temperature microcentrifuge for the next few steps. So aliquot 1 milliliter of culture into 12 pre-chilled 1.5 ml tubes. Centrifuge the culture at 6,000 Gs for 5 minutes and then discard the supernatant. Then gently resuspend each tube by pipetting up and down in 100 microliters of ice-cold 1x wash buffer. Then repeat the centrifuge step. Now you can completely remove the supernatant and then begin to gently resuspend each tube in 100 microliters of ice-cold 1x competent buffer by pipetting up and down. Now the tubes can incubate on ice for 30 minutes, and then they are ready to be transformed with the in vitro reaction following the DH5 alpha competent E. coli transformation protocol on page 16. For the following steps, make sure your reagents are stored on ice during and after preparation. To prepare the Cas12a nuclease, centrifuge the tube of Cas12a protein at a maximum speed for 30 seconds. You will then add 68.2 microliters of Cas buffer directly to the tube of Cas 12A to make 2 micromolar. Now mix by pipetting up and down 5 times and place a checkmark on top of the tube to indicate complete preparation. Now move on to prepare the CRRNA 1364 by centrifuging the tube of CRRNA 1364 at a maximum speed of 30 seconds. Add 48 microliters of RNA buffer directly to the tube by pipetting up and down 5 times. Now place a checkmark on top of the tube to indicate complete preparation. Prepare the 10x in vitro reaction buffer by adding 100 microliters of ultra-pure water to the tube of lyophilized 10x in vitro reaction buffer. Mix by pipetting up and down 5 to 10 times until it has completely dissolved, and then place a check mark on top of the tube to indicate complete preparation. Prepare the HDR-NS oligonucleotide, or ODN, by centrifuging the tube of HDR-NS ODN at a maximum speed for 30 seconds. You will then add 12 microliters of ultra-pure water directly to the HDR-NS ODN tube to make 50 micromolar of the ODN. Again, mix by pipetting up and down 5 times and place a checkmark on top of the tube to indicate complete preparation. Prepare the cell-free extract and ligase mixture by adding 11 microliters of ligase directly to the tube of cell-free extract. Mix thoroughly by pipetting up and down 5 times and place a checkmark on top of the tube to indicate complete preparation. And just remember that the cell-free extract is very temperature sensitive and ensure that it stays cold at all times.